This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 284, recorded on May 9th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today, right here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
I was going to say a smiling Dickson, but you're not smiling as you were earlier.
I'm smiling. Look, I'm smiling.
How have you been, Dickson?
I've been great. Yep.
You're not traveling, right?
Nope.
You were in Banff last week. No?
I was in Banff.
And what was the weather like in Banff?
It was great when I got there. I mean, it was marvelous. I almost decided to take my trout fishing equipment with me, and thank God I didn't, though, because the next morning when I woke up, there was 1.5 feet of snow on the ground. It had snowed overnight, and it continued to snow throughout the day. Not heavy, but, light mist of snow. And the next morning it was still snowing. It was quite remarkable, actually, and everybody else was very disappointed.
Dixon, here the weather is really soupy, isn't it?
It's a soup out there.
You can't see across the river, right?
Nope.
Currently 16 degrees Celsius. Totally cloudy.
Totally.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How's the southeastern portion of Michigan?
It's great. We have really had interesting weather this week. We had pouring, drenching rain. We had 85-degree temperatures yesterday.
Oh, my.
Today it's 67, which is 19 Celsius.
So do you let the weather affect you, Kathy?
Not really. It affects how I get into work.
Yeah, but not mentally. You're okay.
No.
Yeah. The other day I was driving in. It was a bright, sunny morning. It was kind of warmish. And I thought, I never feel this good when it's dark in the middle of winter.
Right,
cold.
Right, that's true. I
think there's really something to this seasonal affective disorder.
I agree.
I just am happy when the sun's out.
Sure.
Mm-hmm.
Also joining us today from a place where the sun always shines, North Central Florida, Rich Condit.
Howdy, everybody.
Hey, Rich.
I know the sun doesn't always shine there, right?
Well, it sure has been great this week, I'll tell you. We've had mid to upper 80s every day and clear blue skies.
Wow.
Crisp mornings, in the upper 60s is just absolutely gorgeous.
Terrific.
And today we have 89 degrees and clear blue skies, low humidity. It's just fabulous. So this is a great time of year.
Uh-huh.
We're not there yet here at this part of the world. Nope. So, Rich, are you immune to the weather? You feel good no matter what?
By the way, 89 degrees is 31 Celsius. I got to get that in. No, as a matter of fact, I remember I did my time in Buffalo.
Yeah.
And I found that especially the delayed spring, which was even later in Buffalo than many places because of the lake effect, drove me nuts. The gray drives me crazy. I just... and when the weather is beautiful, it has a positive effect. So I'm very much in tune with the weather. Yes, indeed.
Speaking of weather and Buffalo, I was at MedImmune yesterday, did a podcast with some of their scientists, and one of them, Ken, had been at the University of Buffalo, or SUNY Buffalo, I guess it is, right? Or is it University?
Yeah, well, so
is it both?
My first PhD student is the sort of director of biological quality control at MedImmune.
Oh, wow.
Really?
Yeah. His name is Schroeder.
Yeah. I didn't meet him.
So you didn't meet him, okay.
But anyway, this guy knew Ed Niles, but he didn't know you.
Okay.
Sorry, Rich. I asked right in the middle of the podcast. Arg. What's that?
Well, that's okay. Not knowing me is not an uncommon phenomenon.
From TWiV, your reputation is truly international.
Right.
I think, Vincent, you were trying to figure out the correct name of it, and because the various SUNYs have changed their names. It's University at Buffalo, the State University of New York.
Okay. I did ask, but, Rich, more people know you now than before.
Oh, that's true.
I mean, that's all of us, right?
I am the beneficiary, big time, of TWiV bumpiness. No question.
I think we all are. Even Dickson.
No especially Dickson.
Well, Dickson, you are famous because of vertical farms.
No.
You were on the Colbert Report.
That doesn't make you famous. It could make you infamous, though.
I watched it the other day. I had watched it before. It was pretty cool. He called you old man when he came out.
He did.
That's pretty.
And he was right. He was right.
I think that's offensive.
No, I didn't take it that way.
You didn't?
Nope.
Dickson.
Old is old.
How you doing, old man?
Doing fine. Sorry, what'd you say?
All right. We have one follow-up today from our last episode on Ebola, which I really enjoyed, by the way. I thought Jens was really good. He talked a lot, but I like when people have a lot of information to convey. Mm-hmm. Did you like it, Kathy?
Yeah, he did have a lot of information.
And we have a follow-up from Robin, who writes, test subjects. I guess we talked about that, testing an Ebola vaccine. For vaccines, for viruses like Ebola, one testing option is to use politicians, economists, and bankers, since they are sufficiently similar to humans.
Arg.
It's a joke, Dickson. Relax.
Yeah.
At one time, German was becoming the leading language for all of science. There are so many German names in anatomy, histology, basic pathology. It was on track to becoming de rigueur globally for all graduate and higher science education until Der Fuhrer derailed it. Yeah, there are a lot of German names and a lot of great German scientists there. And there are many today as well. So they are getting there. They're back on the science railroad there. They didn't include lawyers.
No, I noticed that and decided that it was probably because they're not similar enough to humans.
There you go.
We have lawyer fans and listeners.
I'm sure that they get lawyer jokes all the time.
I know. This is the first one they've ever heard.
No, they are great for telling lawyer jokes, actually.
Oh, yeah?
You remember, Rich, when I came down to give the lecture at your school, I was invited by the law school.
Right.
And I didn't even have to mention the word law jokes. They just started. Spontaneously combusted in front of me.
Today we have two papers, and both of them were provided by Rich Condit. They happen to be right up his virological alley. They deal with the poxviridae. And so I'm going to turn it over to Rich, and I'm going out to lunch.
Back to lunch.
I'll be here, but this is your baby, Rich, so take it away.
Right, cool. I actually even... I'm so pumped here. I actually even made notes.
Yeah, that's what I do when I want to make sure that you get it all, because sometimes you forget.
So I have a scientific paper that's something that I've been waiting for quite a while, and then a commentary that came up in PLOS Pathogens, and I'll do them in that order. I want to start off with a refresher on pox viruses, because we haven't done this for a while. I'll try and keep it brief. So these are large double-stranded DNA-containing viruses. By large, I mean about a 200 gene coding capacity. They're complex. They replicate in the cell cytoplasm, which is a bit unusual. And I guess that's a family. As a family, they are widespread in nature, infecting both vertebrates and even invertebrates. And pox viruses of different species are sometimes very highly species-specific, like smallpox infects only humans in nature, and if you even artificially put it in some other animals, something we'll get to, it doesn't do well. And some other viruses are more widespread, like cowpox is probably the natural reservoir is rodents, but there are transmissions frequently to other vertebrates like cats and people and cows. The most notorious of the pox viruses was smallpox, that basically grew up with humanity and was endemic in most major urban areas, and also caused, superimposed on the endemicity, were epidemics.
Hey, Rich.
Yeah.
I read that a gerbil might have originated smallpox or gerbil pox virus. Do you agree with that? And do you have any sense for when that might have happened?
We're talking basically, I haven't looked this up for a while, but we're talking basically pre-recorded human history. We're going back before the Egyptians, before anybody kept any records. And I think people have been able to trace it back even further than that. And I don't know the data on the notion that it came from a gerbil. It wouldn't surprise me. A lot of these viruses have rodents as reservoirs. At any rate, so smallpox is a nasty disease acquired as a respiratory infection and ultimately blossoms on the skin in these vesicular, that is, pussy lesions that are all over the place, and has about a 30% mortality rate on average.
Why do you die when you die of smallpox?
I've asked that question of the. One answer is I don't know. That's the most common answer, but probably septic shock, basically cytokine storm, everything collapses. So there were efforts throughout history people had a rudimentary understanding of contagion and immunity. They knew that you could get this from other people. They knew that once you got it, you were immune forever, and so they tried to use that knowledge to prevent disease. And some of the first efforts that were practiced for centuries, actually, come under the general rubric of variolation, which means taking some smallpox material and artificially administering it into somebody. And one of the common practices was actually, and this is quite relevant to what we're going to do, actually scratching it into somebody's skin. And you got a really nasty reaction on your arm where it was scratched in, a bunch of blisters and stuff. But by and large, it didn't cause a systemic infection. And so the mortality rate from that process, called variolation, was less than 1% rather than 30%. So even though it was a dangerous procedure, it was a lot better than getting the disease. One of the people who practiced this in England was a physician named Edward Jenner, who also observed that people who had been exposed to a related, what he thought was related, disease called cowpox, another blistering disease with a much milder pathogenesis, were immune to variolation. And he documented this and then did the experiment. He took some material from the lesion of a cow and scratched it into somebody's arm, that is, using his same variolation techniques. They got a much milder reaction and were immune subsequently to variolation. And that's the invention of what we call vaccination. And that then was practiced worldwide, ultimately resulting in the eradication of smallpox by about 1978. It was declared eradicated. So that's sort of pox viruses and their history. The paper I want to discuss is one that's the page proofs are in it. The PDF is online for Journal of Virology. And it comes from the laboratory of Richard Moyer here at the university. He was an old friend of mine. He was down the hall from me. Is there a word that's the equivalent of posthumous for somebody that's retired?
Emeritus.
Because this is published after his retirement. Okay.
Post-professional activity.
The authors are Rice, Adams, Lindsay, Swetnam, Manning, Smith, Burridge, Wallace, McNeil, and Moyer. A lot of authors because this project sort of covered a long range of time and expertise. And it has to do with the notion that the pathogenicity of a virus infection can be influenced by the route of infection. The title is Protective Properties of Vaccinia Virus-Based Vaccines. Skin scarification promotes a nonspecific immune response that protects against orthopoxvirus disease. That's a little hard to assimilate, but I'll get there. And I want to give so I've already said and this is why the smallpox, among other things, why the smallpox story is relevant. There's actually foreshadowing in the smallpox story in that the respiratory disease is lethal. But if you take the same virus and scratch it into somebody's skin, you get a much lower mortality rate. The backstory to this particular paper itself is interesting and has nothing to do with the paper itself. The backstory has to do with a comparison between rabbit pox and vaccinia. Rabbit pox virus is something that has been isolated once from a rabbit colony in Utrecht decades ago, and it was killing all the rabbits. And before the modern age of genomics, it was thought that it was a specific strain of pox virus, distinct from the others, and it did appear to have a much higher pathogenicity in rabbits than other strains that were used. So if you looked at the literature, you got the sense that, for example, rabbit pox in rabbits had a very high mortality rate, whereas vaccinia did not. Actually, subsequently, in the era of modern genomics, we now know that rabbit pox is probably vaccinia that got into rabbits in somebody's rabbit colony. But it is somewhat different than it is a variant of vaccinia. So there are genetic differences between vaccinia and rabbit pox, but they're basically the same family.
What is the natural host of vaccinia?
Don't know. Natural host is medical laboratories. So, I mean, that's a question. The origins of vaccinia are a question all in itself. Okay, I think the best thinking, the current thinking, is that vaccinia probably is derived from horsepox, which is now extinct. And my guess is that if you could go back and to the era when horsepox was around, I wouldn't be at all surprised if you found that it was much like cowpox, where there's a rodent running around with which the virus has equilibrated and doesn't cause a whole lot, but when that rodent bumps up against a horse, the horse gets the disease. That's what cowpox is. Cowpox is not a disease of cows. It's a disease of rodents that cows occasionally pick up because of their interactions with rodents. Likewise, monkeypox is not a disease of monkeys. It's a disease of rodents that gets into monkeys because of their interactions. At any rate, Dick had been working with rabbit pox for a long time. And interested in the pathogenicity of rabbit pox. And since, according to the literature, it looked like rabbit pox had a different virulence in rabbits, was much more virulent than vaccinia, he thought by doing appropriate comparisons and genetic swaps and that kind of stuff, he might be able to identify virulence factors in rabbit pox by doing the appropriate comparative studies with vaccinia. And so, setting up to do these experiments, what they did was they took vaccinia and rabbit pox and tried to reproduce the impressions from the literature that there was a difference in virulence. And what they found was that the vaccinia killed all the rabbits rather than being relatively low in virulence. And this was shocking. Actually, they've got their first supply of WR, the virus that's typically used, vaccinia strain that's typically used, from me, and mine is my own isolate from an ATCC stock that may be a little different than others. And so, the first reaction that we got was, Condit, what have you been doing? This stuff is really nasty. But it turned out that basically any strain would do the same thing. And to make a long story short, it turned out that it had to do with the route of inoculation, because they wanted to deliver a very well-controlled dose of virus to the rabbits, and so they were doing intradermal inoculations. And what that means is delivering the virus. So the epidermis has, going from the top down, there's basically dead keratinocytes, and then going down through the epidermis several layers, you get to a basement layer, which are the dividing epidermal stem cells, basically. And then below that, there's a membrane of fibrous tissue called the basement membrane, and below that is dermis, and then a couple of layers below that is muscle. But an intradermal injection has to do with delivering material below the basement membrane, that is below the epidermis, into the dermis. And you can tell you've done this correct. It's done with a very fine gauge needle, and you can tell you've done this correctly because you get a bubble in the skin where you deliver this. It almost looks like some kind of wheel. That's W-E-E-L. At any rate, so they were doing intradermal injections, and they found that, in fact, in the literature, probably most of the vaccinia infections that people had done were scarification. That's using the same procedure that's used in a smallpox vaccination. And that involves using a thing called a bifurcated needle, which is a stainless steel apparatus that's like a long needle. It's a couple of millimeters in diameter with a flat end that looks like a two-pronged pitchfork. It's got a groove in it that by capillary action holds a couple of microliters of material and then a pointed end. And it's specifically designed so that if you dip it into a preparation of virus, it'll hold in that little groove a couple of microliters of material, and then you can just touch that off on the skin, and the liquid will be deposited on the skin, and then you can poke it with the pitchfork end through the liquid into the skin, and it just barely pierces the skin. You usually poke it about 20 or 30 times until you just barely draw a little bit of blood. And then the virus soaks in through that wound. So that's a very superficial inoculation right into the skin, and that's distinctly different than intradermal.
So you don't get past the epidermis when you do this?
Probably not.
Okay.
I mean, I suppose you could,
if you got blood,
but importantly, you go through the epidermis.
But if you get blood, you're way down.
Actually, you're right. If you get blood, you're going down. You're breaking through the basement membrane. That's correct. Okay, but I think the important deal is that you do significant damage to the epidermis as you're doing this. Right. They didn't want to do that because it's much harder to control the dose. But basically what they found was that when they did these scarifications, whether it was rabbit pox or vaccinia, the animals survived. But if they did intradermal inoculations, the animals died. And in fact, that's the first part of this paper. So let me see. I'm into the paper now. So they started off to look for virulence factors in rabbit pox. And then they discovered that, in fact, genetically, there's no distinction in virulence. Okay? Then that the apparent virulence that what you thought was a difference in virulence from the literature probably is traceable to differences in the way the people inoculated the viruses. So that whole project was gone now. And what emerged from this, is, holy cow, there's a difference here in the virulence based on how you inoculate these animals.
Yeah.
And so the first real data figure in this paper compares the intradermal versus the scarification route. And now they've just said, forget rabbit pox, we're just going to do vaccinia. And basically what you find is that if you give an intradermal injection of 1,000 plaque-forming units, you kill 80% of the rabbits.
Wow.
But if you scarify them, now the dose here is not as clean because they're using a solution that's got a titer of 10 to the 9th plaque-forming units per mil, and they're delivering it with a bifurcated needle, so they figure they're delivering about 10 microliters, so that would be about 10 to the 7th PFU, but you're doing it by scarification, so you don't know how many of those actually get there. But, 10 to the 7th versus 1,000, so it's 4 logs more virus. At any rate, when you do that, you only kill a little over 20% of the animals. So there's a significant difference in virulence. And then they report a bunch of other data like clinical score and temperature and all that kind of stuff. And basically you can see that the animals that are infected intradermally get a lot sicker and die.
I love this one parameter, attitude.
Yeah.
Normal, mildly disinterested, and severely disinterested.
I missed seeing that, but yeah, that's great.
Figure 1 is all of the details about how they award the clinical scores, so that's pretty interesting.
And a disinterested rabbit, that's just Alice in Wonderland.
I used to work on rabbits a lot to make antibodies in them, and of course intradermal injections are the thing to aim for with regards to complete Freund's adjuvant. And in fact, we ended up not being able to use complete Freund's because of the amount of tissue damage it induced. Mm-hmm. But these were multiple injections, right? And so you'd come back the next week to try it again, and this rabbit didn't want to see you.
Yeah.
Right.
This was an aggressive rabbit. A Monty Python rabbit, I would say. Initially,
rabbits are pretty amiable creatures, aren't they?
Yeah, but look what we were doing to them, unfortunately. Yeah. So, absolutely.
So then the next question becomes, in comparing these two routes of inoculation, what happens? And so there's the first, the assay that might be is amusing, if nothing else, is spread. So do the viruses spread to the same extent. And one of the interesting things about the rabbit model is that this is the pox virus model in rabbits is that this is really straightforward to assay because you just look at their ears. The ears are relatively translucent, and you can see a bunch of blood vessels and stuff. And if there's spread of the virus systemically, you get pox on the inside of the ears. So all you got there's this picture there, figure 3. All you got to do is hold up the ears and look for pox.
Nice.
And basically, they saw pox in animals infected by either route. So you can see the primary lesion is in the second row there. The primary lesion in the intradermal is a lot nastier than ultimately than in the scarification. But you got secondary lesions in the ears in both cases, and maybe even a little more in the scarified rabbits, and they're thinking that's because they last longer.
They're infected for a longer period of time, so that secondary stuff can be developed and we can see it.
Right.
Okay.
So then they assay a bunch of other tissues to look for spread, and they basically see that both viruses spread systemically. There's maybe a little different, maybe a little less little less in the liver from the scarified animals, and ultimately less in the spleen from the scarified animals. But I think importantly, in both cases, there's virus spreading throughout the animals, and yet the scarified animals are recovering. And so then they look to see whether there's what the histology looks like at the site of the infection, whether there's immune cell infiltration. And yes, there's immune cell infiltration regardless of how you do it. But importantly, if you look at animals that are mock scarified, that is, you just take a needle that's got phosphate buffered saline in it, no virus, and do the scarification procedure, you get an influx of immune cells. So there's an inflammatory response just to the scarification itself. And so basically, and there's some other details of the immune response that they go into, that there are some subtle differences in the response. But basically, I think the bottom line is that it appears as if the scarification is doing something all by itself. And so then comes the experiment, and as far as I'm concerned, this is the real meat of the whole thing. And that's, I guess, well, the readers aren't going to be, the listeners aren't going to be able to see this, but figure 5, they have an experiment that they call ring scarification. So here, excuse me, they want to test whether scarification itself is actually doing anything. So what they do is they do both an intradermal injection and scarification with just PBS, just buffer, no virus. So they either do an intradermal injection and then in a ring around the intradermal injection take a bifurcated needle and just do a scarification, or they do it the other way around. Do the scarification in a ring and then an intradermal injection in the center of the ring. And what they find is that if they do the intradermal injection first, and then the scarification with a blank needle around the intradermal site, it looks just like an intradermal infection, and 80% of the animals die and they get sick and all the rest of that. But if they reverse the procedure and do the scarification first, and they're doing these procedures within 2 minutes of each other, do the scarification first and then the intradermal infection, now only, 25% of the animals die. They get a much higher survival rate, and the animals, they're still pretty sick, and they're sicker than if you just did scarification, but they are measurably less sick than the other animals. So the scarification itself is doing something to influence the survival.
Rich,
messing around with the skin.
Don't mean to interrupt you, but I don't have the paper directly in front of me. How many rabbits per group?
There's
12, I think.
I would have to look up in the map.
Yeah, 12. Yeah. I think it's 12.
12 for the scarification, and then intradermal. For the other way around, they use 6.
Yeah, right.
Is that good, Dickson?
That's for the experiment reported. They've done this several times and with different doses. Okay, so scarification of the skin is influencing the outcome of the intradermal injection, somehow apparently stimulating immunity. So then they do some experiments to figure out exactly what it's doing. In particular, what they do is they take samples from of the site of inoculation from either the intradermal or the scarification, and they make RNA from those and analyze it on a specialized chip that they have that was made by another lab here at the University of Florida that's rabbit genes, and look at the gene expression profile. And they have all of the data reported in a supplementary table, but in one figure they report a heat map of the genes that are most prominent in allowing you to distinguish between an intradermal and a scarification. And you can see that these are basically mirror images of each other. There's the bottom line is there's a big difference in some of the rabbit genes that are expressed in response to an intradermal or a scarification. So there's something different going on there. They also report that they did some control experiments where they did an intradermal injection on one flank and a scarification on another flank, and that did not have any protective effect. The scarification has to be around the intradermal infection. So the bottom line is that agitating the skin has an effect on the immune response, apparently. That ultimately, in this case, gives a much better protection of the rabbits from the infection. Their hypothesis is, and there's some evidence to some studies to back this up, apparently keratinocytes, among other things, in the upper layers of the skin are big bags of cytokines. And if you disturb them, they release a bunch of cytokines and stimulate an immune response. And the idea is basically that the cells in the skin that are there for the purpose of detecting pathogens get a much earlier and stronger signal and run off to the local lymph nodes to tell them that there's something up. And in an intradermal injection, you bypass that, and so you don't get that early warning system.
Wow.
So the skin is immunologically an early warning system.
So that's the part of the defense because your skin's always getting scraped, right?
Absolutely.
And so this is to take care of that, right?
Sure, right, absolutely. Sure. It makes perfect sense.
And
probably the same goes for the mucosa.
Right. Right. And mosquitoes bypass this.
You bet.
They go right through the epidermis, right?
Oh, yeah. They go for the blood.
So among other things, this made me think of we've covered recently a couple of papers talking about delivering vaccines.
Right.
These patches, right into the skin, and about how you need a much lower dose of the virus, of the vaccine to do that, and you get a more robust immune response. And so I think that this is, well, first of all, it's fascinating. And second, I think it really is of very practical significance ultimately.
So these vaccine patches that we talked about, they have little needles on them that probably are scarifying the epidermis.
Absolutely.
And it's giving this cytokine release, so that it enhances the...
Interesting.
Wow, so cool. I mean, one of the things that the skin has a lot of is antigen-presenting cells, and I bet they're brought there by these cytokines.
The dendritic cells must be all over the place.
Absolutely.
So, Rich, if you took...
And they run away really fast, to deliver their message.
Very fast.
Rich, if you took the smallpox vaccine, instead of scarifying, I don't know how you could do this in a rabbit. If you put it intradermally, would it be lethal?
Well, that's what vaccinia is.
It's basically the smallpox vaccine.
Sure.
Okay, so yes, it is lethal. The next question is, what happens if you do an intradermal injection of that in humans? And I'm not going to do the experiment. Well, it wouldn't work on me anyway, because I've been vaccinated so many times.
Yeah, right.
But I have a hunch that if that was a bad thing, we would know about it.
Yeah, okay. I'm sure that there are times when people go too far in with the bifurcated needle.
However, if you look back historically, remember I told you, this is one of the reasons I gave all this background, remember I told you about variolation? Mm-hmm. Apparently there were good variolators and bad variolators.
Huh.
This was a technique that you wanted to go to the good guys, not the bad guys. There were some people who had a much higher mortality rate in their variolation practice than others, and there was a lot of theory about how you actually applied the technique of variolation and what the outcome was. And I will bet you that it has to do with how deeply you applied it, or how much or how little you disturbed the epidermis in the process of variolating.
Fascinating.
I have to tell you what D.A. Henderson told me. I interviewed him a couple of weeks ago for our textbook, and D.A. Henderson, of course, was the organizer of the smallpox eradication campaign. He told a great story of that, but that'll come out eventually. He said that in the early days when they were doing the eradication campaign with the bifurcated needles, they were told, do not draw blood. That's not good. You'll get a poor take. Huh. And he said, but they found that now and then they would draw blood accidentally, and they learned that gave a better antibody response when you drew a little bit of blood, so that they ended up doing it a little harder so you would get some blood out. I don't know what the relevance of that to this is, but you said, Dickson, if you draw blood, you're going into the dermis, right?
Yeah.
You're going into the...
Actually, you're going even through the dermis.
You're going through the dermis into the vascular bed, as it were. Yeah.
I've always thought that the drawing blood issue just let you know that you had indeed pierced the skin. Okay, so it's a direct sign that you've made it through.
Right.
I think you could just watch the vaccinee's face.
That's true. That's true.
So there's several things I like about this. So first of all, I like the basic conclusion. I've watched this evolve over a period of time, and I just think it's so cool. I think it really emphasizes how important the skin is in immunity, and I think it has practical implications down the road. I think it's, it tells us, I knew this, but pox viruses are cool. And still cool as research tools. And it's also, I think, a terrific lesson in what we talk about all the time, and that is serendipity. Right? Because this started out, well, let's look at the difference in virulence between these two viruses, and it turns out there wasn't any. But figuring that out got them onto this route of inoculation thing, which is something that they never would have investigated otherwise.
So my question is, how broadly applicable is this? Now, this vaccine replicates very well in these outer layers, right? But not all vaccines do that. So like polio vaccine is given intramuscularly, the inactivated vaccine. Would that benefit by scarification? Maybe not, right? I
don't know. I don't know. You'd have to do the experiment. As I recall, I'm thinking of the TED video of the guy who has, I and I forget the names, I'm sorry, the guy who was talking about these patches that he's made with microneedles.
Yeah.
I don't know what viruses they've tried in that. I think flu was one.
Yeah, flu was one of them, which is currently done IM. So, yeah. I mean, this paper deals with virulence as a function of inoculation route,
right.
But the issue is, for a virus that's inactivated, that's not an issue. What is the issue is, would you get a better response if you did this as opposed to putting it deeper?
So what is the route of transmission for smallpox?
It's usually aerosol.
Exactly.
And so it's acquired as a respiratory infection, probably infects respiratory mucosa.
Sure. So that completely bypasses this mechanism.
Yeah, I know, but I'm just thinking of inducing good protective immunity by getting this great epidermal cytokine response.
Right. So there's another parasite, leishmaniasis, dermal leishmaniasis, that has a history. Before we even knew what it was, the Chinese were scarifying for that. So maybe, just maybe, some of the recovering smallpox victims were mistaken for dermal leishmaniasis patients or people suffering from that, and maybe the scarification for smallpox started earlier than we think.
Could be.
Just a thought.
The other thing that strikes me about this, so the lesson here is something I teach my students in virology, that when you talk about virulence, you have to be very specific about virus, delivery route, dose, and animal, and all that.
Sure.
And remember the ferret influenza H5N1 story, where initially we were told that the aerosol passage, or the H5N1 that travels by aerosol among ferrets, was lethal, but it didn't tell us that it's only lethal if you give it intratracheally. But when it passes through the air, it's not lethal for ferrets. So route is very important.
Oh, yeah. Some recent findings from the malaria vaccine work also verifies that as well. So
what will be interesting to discover if the experiments get done is, I mean, you raise an interesting point with how would this work with other viruses, in particular viruses that are not necessarily, I mean, there aren't many viruses that you acquire through superficial lesions in the skin.
Yeah.
Right? But this may be a way of sort of, if you like, fooling the immune system with a vaccine. You may be, even if this is not a natural route of infection for a given virus, you may be able to take advantage of the fact that the skin does this to deliver a virus through the skin and get an enhanced immune response.
I think basically for an inactivated vaccine, that makes perfect sense.
Right. I mean, polio's that way, isn't it?
Yeah, polio's, the inactivated version would make sense. I'm thinking of other attenuated vaccines where they have a special cell that they have to replicate, and this wouldn't particularly work. But for inactivated,
I mean, like rabies.
Yeah, rabies or inactivated polio or inactivated influenza vaccines. Yeah, they might benefit. So someone should do a trial.
Basically, the way Moyer describes this, his bottom line is scarification is kind of an adjuvant.
Yep. Perfect. That's right. We don't need no stinking Freunds.
No. That's right.
So, I mean, if you just scarified your rabbits, maybe you would need to use complete Freunds, Dickson.
There you go.
Well, they outlawed complete Freunds because of the adverse reactions that it was eliciting with the tuberculosis organisms that they put in there. So they eliminated. Well, then we had other adjuvants after that, but that was.
So this is really a nice story. So this is his last story, basically?
No, I think he's got some other stuff still piled up that will come out. I've thought about this, Kathy. Didn't you say that John Holland kept publishing after he retired? I think, and I can see where this might happen to some other people that I know. You don't necessarily get to wrap up everything real tidily.
Like you, Rich?
Like me, maybe.
I think it's really nice. I'm going to incorporate this into my course next year. It's perfect.
Cool.
It's really one of those
really cool.
illustrative, very clean observations. Sure. I like it.
Sure.
I hope Richard listens to this episode.
He will. I've talked to both Dick and Amanda, the lead author, in preparing for this.
Nice work. Very nice.
Okay, so the other thing I came up with, we ready to move on?
Yep.
The other thing I came up with is a commentary that was recently published in PLOS Pathogens called "Are We There Yet? The Smallpox Research Agenda Using Variola Virus." And the authors are Inger Damon, Clarissa Damaso, and Grant McFadden. And before I get into that and even a little mini bio on the authors, I'll give the backstory for this. Now, this is a topic that we've, and actually we've even, I think, discussed commentaries like this in the past. This is an update, if you like. So the backstory is that, as I said previously, smallpox was declared eradicated in 1978. And when the eradication was declared, then the question becomes, what do we do with the smallpox that is left on the planet in various research laboratories? Actually, this was stimulated by at least one lab accident that happened after the eradication was declared, where somebody in a research lab, actually somebody peripheral to a research lab, became infected with smallpox from that lab and died. So that's not good. And on the heels of that, though, I think there may have been plans to do this anyway, they asked any investigators who had any smallpox to destroy it. And the remaining stocks were consolidated in 2 repositories, one in the public health laboratory in Moscow, right, and the other in the Centers for Disease Control. And so there are repositories of smallpox virus under high security containment in both of those facilities. And as we've discussed before, it's important to understand that we're not just talking about 1 or 2 vials. We're talking about hundreds of vials representing hundreds of isolates, some of them redundant of each other, but we're talking about potentially different strains of different virulence and from different locales. So these are that's not a genetically homogeneous collection of viruses.
And not all of them have been sequenced.
And not all of them, about 10%. We're talking about maybe 500 different samples at least.
Wow.
And probably only 50 have been sequenced, maybe 10%. Okay? And soon after all of this happened, the question arose as to, why do we need this stuff anymore? Let's just get rid of it. And this is not too difficult to understand, in particular coming from people who were instrumental in the smallpox eradication campaign. They were boots on the ground. They saw what a horrible monster this actually was, and it was a tremendous effort to get rid of it, and they don't see any reason for keeping it around. As a matter of fact, it's potentially hazardous, so let's just finish the job and get rid of it. And so this has been on the agenda, whether or not to get rid of the existing supplies, for 20 or 30 years now, and it keeps coming up to the World Health Organization. And so a little bit about the organization of the bodies that consider this. There are committees appointed or convened by the World Health Organization to consider this, and the one that has been active for the last 15 years or so, that is most active in this, is the WHO Advisory Committee on Variola Virus Research, the ACVVR. And they are charged with supervising any, actually the decision was made at one point, I think in the early 90s, to actually even encourage or at least allow some continued research with variola virus to understand a few principles about its pathogenesis. And then, of course, 9/11 happened. And the whole biodefense flap started, and there was motivation to make sure we had vaccines and antivirals to deal with smallpox, it being sort of number one on anybody's list of a bioterrorist agent. And so the U.S. government, for example, said we need to have a safe vaccine, and the vaccines that were available, the stocks were low, and they wouldn't pass FDA muster now, and they also had adverse effects, so we wanted second, third generation vaccines, and they also wanted 2 antivirals. And to make sure that these things were working properly, you needed variola virus as a model virus to test the efficacy of these things. And so, in fact, variola research was not only allowed, but in some circumstances encouraged. And the WHO Advisory Committee on Variola Virus Research was convened to basically review the research on an annual basis, at least, and see what was going on. And there is another committee who then reviews even all these reports. The WHO committee is mostly virologists and with a heavy complement of pox virologists. There is another group that even reviews those reports called the Advisory Group of Independent Experts to review the smallpox research program. This is another WHO organization. So that gets abbreviated AGIES, AGIES. And these groups on a regular basis generate reports that are then handed out to what's called the World Health Assembly. The World Health Assembly is all the member nations or committees from all the member nations of the World Health Organization. So it's sort of the legislative branch of the WHO, and they convene and come up with some sort of resolution about their opinion about this, and then pass that resolution off to the World Health Organization, which then, I guess, issues some sort of directive. Up until now, the World Health Assembly has not recommended, its most recent recommendations over the last few times it's considered this has been to delay nuking the last repositories of smallpox. Like they read the reports of these advisory committees and said, it looks like more work needs to be done with variola, so although we would like to nuke it eventually, we're not ready yet. And in fact, the last time this came up, it was a year or so ago, right before the World Health Assembly met, the United States government, I forget exactly in what form, whether it was the Surgeon General or what, issued a statement that said that if the World Health Organization says that we are supposed to nuke these remaining stocks, we're not going to do it. Okay? So that's what McFadden referred to as a shot across the bow, which basically preempted, I think, and was probably intended to preempt any action by the World Health Assembly, because what are they going to do? If the U.S. has declared they're not going to cooperate anyway, sure, it would create a pretty awkward situation if they voted to get rid of it. At any rate,
I'm sure Russia wouldn't cooperate either.
Well, that's something that we can discuss here. So at any rate, the World Health Assembly is going to meet this month to consider the most recent reports of these 2 advisory groups, the AGIS and also I can never remember these acronyms. The ACVVR.
Just remember VR, you know.
Right. Okay. There you go. Okay, I can remember that. And these most recent reports, in fact, are closer to recommending that it's time to get rid of smallpox than any previous reports. I think that's fair to say. And the reports are available as links from this commentary that I've that we're going to discuss. And basically, they say the two reports differ a little bit, but in the most critical areas, they say that the majority, and they're very careful to call it a majority rather than saying it's unanimous, that the majority of the committee members don't think that it's any longer necessary to keep smallpox around to accomplish certain research tasks in the interests of the public health. And so the thinking is that if that language goes forward to the World Health Assembly, that it is not unlikely that the World Health Assembly will make a recommendation that we get rid of the rest of the smallpox stocks. Okay?
Mm-hmm.
So, in the face of that, this commentary was written by Inger Damon, Clarissa DeMasso, and Grant McFadden, who are advisors to the ACVVR.
Mm-hmm.
They are not actually the ACVVR has 20 members, and then there's another 20 scientists who are advisors to the ACVVR, who are very close participants in this whole thing. And Damon and DeMasso and McFadden are three of those individuals. And basically, I hope I'm not overstepping my bounds here, but I would say that this reads more or less, I would call it almost a minority opinion. If the majority of the individuals on these committees are saying we don't need to keep these things anymore, these three individuals are saying, no, we still need to have these around for a while. So I would call this a minority opinion. And I think their first paragraph really by the way, they are not only advisors to this group, but Inger Damon, in particular, is the chief of the poxvirus and rabies branch of the CDC and director of the WHO Collaborating Center for Smallpox and Other Poxvirus Infections at CDC. Okay, so she's really extremely knowledgeable about all of this, and it has her finger very much on the pulse of poxvirus research and smallpox virus issues. And she's been at the CDC for a long time. I don't know how relevant it might be, but Grant McFadden is also the co-editor-in-chief of PLOS Pathogens, where this thing is published. So I just want to read their first paragraph, which summarizes the whole thing. Despite significant advantages, despite significant advances, there is more work to be done before the international community can be confident that it possesses sufficient protection against any future smallpox threats. The current World Health Organization approved research agenda for smallpox has been tightly focused by the interpretation that research, quote, essential for public health, unquote, equates solely to applied research related directly to the development of new antiviral drugs, safer vaccines, and better diagnostics. Despite considerable advances in this direction, we argue that the research agenda with live variovirus is not yet finished and that significant gaps still remain. So they're arguing, they're arguing, first of all, that the definition for what we need the virus for is too narrow, that it's focused specifically on development of new antiviral drugs, safer vaccines, and better diagnostics, and the existing virus strains have utility beyond those narrow goals and should not be destroyed for that reason. And most notably in that case, smallpox is very specifically a human pathogen. And we don't know what the determinants of that specificity are. We're clueless. Whereas other pox viruses can infect or can be specific for other organisms or can be relatively nonspecific. And so in terms of understanding viral pathogenesis, understanding the determinants of that specificity and virulence would be a very valuable thing to know. But these authors even argue that even within the narrow definition of the World Health Organization, for example, research for antiviral drugs, that we're not necessarily even done with that yet, because the two drugs that are on the front line, CMX 001 and ST246, are not actually licensed, and they could even fail licensure. It could be that in the future it would turn out that there's too much resistance to those, and you need some other drug. So they're not convinced that even those narrowly defined things, we're done with them yet. And so they argue that these stocks should not be destroyed, and we should retain them for future research.
So, Rich, let me ask you, I used to run a course here with Steve Morse called Emerging Infectious Diseases, and we talked about this a lot throughout the years that I helped teach that course. And Steve actually had taken some trips to Russia because he was on the advisory board for DARPA. And they were really interested in knowing how they knew that they had X number of vials and which was in and what was in each one. If only 10% of all the stocks of smallpox have been sequenced, what has stopped us from just sequencing all of them, at least to have a permanent record of what's available out there?
That is one of the ongoing projects.
I mean, isn't that an easy thing to do?
Well,
come on,
time.
You get a whole human genome in a day.
Well, it's straightforward. I think
that's what I mean.
It should be straightforward.
So why haven't we done that at least?
That is in progress, but it's not done. Okay, which is another reason for not getting rid of these things yet.
Right. But in fact, the other thing he learned, by the way, in that interview that he took in Moscow, he was in Moscow, was that they're not actually sure of how many vials they had. So they're not sure if any of them are missing. And that's the biggest fear. This comes up all the time, and that is, how do you know that these bits are the only ones out there? So it's like spy versus spy type of thing in Mad Magazine. It's exactly like that. Is who's going to go first, and then whoever goes first, well, that's too bad because we decided not to go first. So now you're at our mercy because we've got all the smallpox and you have none.
Relative to your first question, there's a table in one of these reports. That is, let me see, list of variola virus material used for genomic sequencing without propagation.
Yeah.
135 removals of 135 strains, 70 of which were used to completion. So I think that, I don't know what the strategy is here. There is a strategy to reduce the number of stocks. That is, take the last of a vial and sequence it, and then get rid of the rest of that. Now, whether that's how they choose which ones that are going to be deleted from the collection, whether they're redundant of some other samples or something, I don't know. But out of a little over 400 samples at the CDC since they last did this, they've actually depleted 70 of them. In the process of this sequencing project. So they have in mind getting rid of at least some of these as they conduct this sequencing project.
Sure. I think these documents, by the way, are terrific.
Yeah, they're
they
good.
are full of great summaries. So Dennis Ruby has a little bit on his antiviral ST206. Is that it?
ST246.
246. And all kinds of other summaries of what's going on. It's really, really nice that they put it out there for everybody.
Yeah, the executive summary on the whatever it is, VR, right?
ACVVR.
The
VR,
executive summary on that is very good. And then there is a summary at the end of the Aegis thing that is also very good, where they have 5 points about how the members conclude that there's no need to retain live variola virus for the further development of diagnostics. Okay, and then for sequencing, and then for animal models, for vaccines, for antiviral agents, and they note at the bottom that recommendations 1, 2, and 4, but not 3 and 5, are in line with the conclusions of the other committee, the ACVVR. So it's really actually very readable and very straightforward.
Yeah. So that's interesting that you say that they don't need virus for diagnostics, but McFadden et al. In this article argue that you do, that the diagnostics aren't good enough.
Right.
So there's dissension here.
There is
clearly.
dissent. And in fact, if you look at the executive summary of the ACVVR, let me see, I've highlighted these. With respect to the issue of whether you need diagnostics, it says the majority view within the committee was that there was no need to retain live variola for this purpose. Mm-hmm. It's not unanimous. Mm-hmm. Okay?
Yeah.
They're very careful to use majority. Second issue, needed for vaccines. The majority said there was no need to retain it for vaccines. Third issue, whether live variola was needed for further essential for antivirals. And that one, it says the majority within the committee was that live variola was needed for further development of antiviral agents. Mm. So in that third issue, even the ACVVR thinks that we're not done. The majority, however, there's some who thinks we are done. So it's not an open and shut case. And the commentary basically comes from individuals who believe that in probably all of those realms, we aren't finished yet.
Right. I mean, if you look back at the anthrax episodes, all right, where the hell did that stuff come from? Who did that? How was it done? It took a long time to figure that out, right? And that's was something less volatile than this. Mm-hmm. So the big paranoia that's created by I don't trust you, you don't trust me, is that we will just stall and stall and stall and stall. And that way we can maintain these stocks forever.
So Dickson,
some people may take that view that's what we're doing.
What do you think should be done?
Well,
destroy or not destroy?
No. I think you can't unilaterally destroy. You have to cooperate. You have to put all of the samples together in one place. First of all, it has to be in one place.
Well, that's not going to happen.
Well, that's what you asked me what I was going to do. I'm going to tell you what I would do.
No, I gave you two choices: destroy or not destroy.
Well, I'm going to get to that.
Okay.
Because you can't destroy only your stocks and depend on the other guy destroying their stock. You can't do that. So this has to be a WHO committee that establishes a central place for all of the stocks of smallpox, all the legitimate recorded stocks of smallpox. You put them in one place, and now you can make your decision.
So during the transport, that's a perfect time for someone to steal.
I know.
Some of them. It's not happening.
Did that happen with nuclear warheads when they agreed to downsize?
Well, they didn't eliminate entirely.
No, but they sold them all to us.
Okay.
We bought the warheads and turned them into nuclear power.
And if you're going to move them to somewhere, how would you know that both places moved everything they had?
Of course.
Kathy,
no, it's just,
precisely.
It's never, so, yeah.
You'll never eliminate the paranoia in this.
Yeah. Just leave them there.
And it is also...
Unplug the refrigerators.
It is also now technologically possible with the sequence, theoretically possible with the sequence out there to reconstruct at least some of these strains anyway.
Sure.
Rich, what would you do? What do you favor doing?
I do not favor nuking them at this point in time. There may come a time, okay? I don't feel as strongly about this as I used to, okay? But there may come a time when it's obvious. But what always holds me back is that once you get rid of these, it's an irrevocable decision. You can say, yes, that you could reconstruct the viruses, but in fact, the sequence information is somewhat limited. There's a technicality here, and that is that when you say that we have genomic sequence, that is most of the genomic sequence. These viruses have a funny genome structure. It's double-stranded DNA with a hairpin end. Okay, so it's DNA all the way around on both ends. So it's like a self-complementary single-stranded circle. Self-complementary single-stranded circle that snaps back on itself. It's got these hairpin ends. When people do sequence, the hairpin ends are really hard to sequence, okay? And typically those are not done. That doesn't have coding coding sequences in it, but it's not, I don't think, completely clear how related the hairpin ends are from one strain to another, or how that might affect the properties of the virus. And if I understand it correctly, there's hairpin end sequence for only one smallpox strain. And then there's genomic sequence for, 50 or so others. And so if you nuke all these, you're getting rid of genetic information that you are not going to be able to recover. And I'm not ready to do that yet, okay, because that's an irrevocable decision. You may regret that down the road. Plus, I believe that these things can be held safely, right? So far, there's not been a problem. I don't anticipate there's going to be a problem down the road. So I don't see a threat with keeping them. I do see a potential downside to getting rid of them. That having been said, I understand if nothing else, the emotional and political motivation to get rid of these things. But I think it's problematic.
What did D.A. Henderson say?
Oh, he's a driving force in getting rid of them.
Right. Because that's what his job was before.
That's what his job was, and he wants to finish the job. I don't blame
exactly.
Yeah, I didn't ask him that. Kathy, what do you think?
I pretty much would align with Rich. I think for now there's no point and there's no practical way to get rid of them.
Yeah, I think I would agree with that. But it's funny, Rich, you say you don't feel as strongly, and I feel the same way. I used to be very strongly against keeping these forever, but now I could see at some point not. But I think we need to finish what needs to be done as outlined in this article, so we shouldn't do it now.
Does that relate to the fact that since your original feelings, we have developed many, many new antiviral drugs that actually work? So the hope is that there'll be an antiviral drug that'll easily cure smallpox someday, too.
I don't think so. I just think I don't know what the reason for the change is.
Yeah, I mean, are there Richard? Are there any good pox virus antivirals?
Yeah, ST246 is terrific.
It's terrific. So
as far as we know, I mean, it's never been tested in humans, but in every other animal model where it's been tested, it works very well. But now there is the potential for resistance. You can get resistant mutants to that.
Yeah, sure.
So on a large scale, I don't know. Now this other one is a cidofovir derivative, CMX001, I think it's called. That is pretty good as well. I'm not sure that I'm not convinced that it is as good as ST246. But what the mandate was for two anti-pox virals that work by different mechanisms, and that is a DNA replication inhibitor. But I think there's room for more.
Right.
TWiV 270, we talked with Dennis Ruby about ST246. Mm-hmm. Go check it out at Dickson. Okay, go read it.
Okay.
And you'll get a quiz on Monday.
I will be, forget about that.
During this discussion, ACVVR, I just realized my initials can stand for virus research.
There you go.
True.
I just didn't think of that before. That's why I'm a virologist.
Must be.
Pose. Let's see, was there... Oh, I want to put up a poll on this issue. I want to see what our listeners think, and I was thinking, tell me what you think. Should we destroy remaining stocks of smallpox virus? Yes, no, not this year, but in the future.
Wait, you have to define who we is.
I'll take out we. Should remaining stocks of smallpox virus be destroyed?
All remaining stocks.
Should all remaining stocks of smallpox virus? And you like the choices? Yes, no?
All known remaining stocks.
Okay.
That's right. Officially known.
And yes, no, not this year, but in the future? You like that? Or should
I think I like leaving a third option in.
All right.
My guess is that, this is the way with most political processes, that the tabling option gets used most frequently. But I mean, that's
absolutely,
it's not an unreasonable thing to do in this particular case. That's my opinion.
I have a geographical point. The stocks are held in Koltsovo, which is in Novosibirsk, Russia, which is pretty much straight north of India, not Moscow.
Mm-hmm.
Okay, sorry.
That's right.
My bad.
As far as we know, right?
As far as we know.
The ones that are reported in the ACVVR report, executive summary.
Yeah. Right. Nice job. Thank you, Rich.
You're quite welcome. My pleasure.
Good stuff.
Thank you for the opportunity to do that. What a wonderful opportunity to be able to, talk about this stuff.
It's remarkable. I think I've said this before, but I'll say it again because it's been a while since I've been on the show. It's remarkable how good we are at extincting species that do us no harm. and how difficult it is for us to get rid of something that does us great harm.
I think that this would be the first deliberate destruction of
I don't think so. No.
species.
No, that's
Deliberate?
not right. The dodo bird was deliberately destroyed.
Is that right?
Absolutely. They ate them all.
Oh, okay.
But
And
they know they were destroying them?
Yeah, sure. Then there's the auk. The auk. I believe it was a very large flightless bird in the north, in the Bering Sea or something. And they ate all those too. They ate the fat. They ate the passenger pigeons. Come on, they ate a lot of things.
Well, and then there's this talk about that they might be able to bring back the passenger pigeons, and then other people are saying, but do you really want to bring them back? They're going to come and poop all over people's cars.
No, but they were delicious. They were delicious.
Yeah.
You're absolutely right. You're absolutely right. But we purposefully extincted a whole bunch of things. We did, including some people who lived on an island off the coast of Australia. We genocided an entire island, and the Tasmania island as well. So we're not such a nice species when it boils down to it.
No, we're not.
Oh, yes, I see, Kathy. I misspelled wheel.
Yes, don't worry about that. Don't worry about it. Rich.
That is me, of course.
W-E-A-L.
W-H-E-A-L.
Okay, good.
I thought so, but I didn't want to interrupt him. All right, let's do some email. We have 3 email having to do with the Tamiflu episode of a couple of, that was not the last one, but the one before. So I'd like to read all 3 and then have some comments on them. I will take the first because it's short. Christoph writes, "Hi, Vincent et al. I was wondering if you had read this blog post by Mark Crislip." And Mark Crislip, of course, is an infectious disease physician out in the Northwest who has a podcast of his own called the Puscast, the Infectious Disease Puscast. I listen to him. He's very good. He's very opinionated. So he has also a blog. He writes at a blog called Science-Based Medicine. So he wrote something called Cochrane Reviews, the Food Babe of Medicine question. I would love to hear you and the TWiV team discuss his post. All right. The next one is by Igor. Please, Kathy, can you take that one?
Sure. Dear Twivers, please forgive the delayed follow-up. After listening to your episode number 282, I posted a short comment about the Tamiflu paper in my blog, and one of the readers pointed me to the following 2 blog posts that discuss the same paper. One of those is to the same blog post by Mark Crislip, and another one is also on that science-based medicine blog, and offhand I don't remember that author's name. Anyway, they are quite interesting and put a bit of a different light on this review for me. The first post is shorter and more to the point. Let me quote from the conclusions. What the news coverage and the authors seem to be ignoring is that Tamiflu is demonstrably and meaningfully effective at preventing the spread of the infection. At the same time, there's some real-world evidence, albeit with limitations, that suggests that oseltamivir may provide a slight survival advantage in the very ill. The seriously ill and those at greater risk for flu complications have not been included in the analysis. What the analysis says is that the drug is of limited benefit to healthy people during the relatively mild seasons of flu. One can't generalize from this data to the potential benefits of the drug in the case of a more serious pandemic or to all populations. The second post is a bit more rambling, yet still very interesting. It can be summed up with the following quote. Just because something is labeled as a systematic review does not mean it is any good. We have to be just as vigilant now as ever. Even a review with a Cochrane label does not make it true. The author then goes over this paper and some previous Cochrane reviews to show some substantial flaws in the ways they were done or interpreted. Finally, both posts on the same blog but by different authors point out that 2 of the authors of the Tamiflu paper have a history of downplaying the seriousness of flu, going as far as suggesting that vaccinations against flu should stop. After reading these posts, things don't seem so clear-cut to me. Certainly more studies are needed on the benefit of Tamiflu for treating serious flu cases, and perhaps we should not stop stockpiling the drug right away. Best regards, Igor.
Okay. And Rich, can you take the third one?
Joseph writes, Dear Doctors, I wanted to follow up on the Tamiflu Cochrane review discussion from episode 282. I can't say that the results of the meta-analyses surprised me, as I am pretty convinced that these types of drugs have only modest effects in treating influenza infections. For one, and maybe this is naivete on my part, I am always slightly skeptical that antiviral or other anti-insert your pathogen of choice here drugs that need to be given early during infection will have a significant impact on disease progression. Actually, I find that too many publications investigating the effects of compounds/drugs on pathogen replication, etc., either A, ignore timing, or B, incompletely discuss the issue of timing. That is not to say that these aren't really interesting, good, and important studies, just that the timing can make a big difference. One thing that I thought about during your discussion was that for individuals who were critically ill from influenza infection, could that small time decrease in alleviation of symptoms be helpful? In other words, how would ID doctors view this report/data? Over at Science-Based Medicine, Dr. Mark Crislip takes a look, although it does seem that he is not a big fan of some of the authors of the paper anyhow. It's definitely worth a read. Joe.
All right, I have a couple of thoughts. So the article by Crislip, Crislip is opinionated and skeptical. He doesn't like meta-analyses in general. And, we've talked before about their shortcomings, and I fully agree that you don't accept every one. But as far as this one was concerned, it seemed that it was done well. Okay. Now, his feeling is he has some anecdotal data where he says, I've seen serious flu, and I think it makes people live. But we can't use any of that. It's anecdotal. It's not a properly controlled trial. So despite what he or any ID doc feels, nothing of that is of any use in real-time usage unless there's reason to believe that it's going to work. Now, he goes on to criticize previous Cochrane studies, and that's fine, but that's not really relevant. The point is this study good? And in fact, in this study, which we discussed, they looked at randomized controlled clinical trials, which, as the other article points out, is mainly going to look at pretty healthy people. So in reasonably healthy people, it has a marginal effect. The other blog post raises the issue of, what about really sick people? You can't study those in a randomized clinical trial because you can't withhold treatment in the control population. So you have to do other kinds of studies. They have been done, and in fact, there was an accompanying article in the same issue as the Tamiflu paper and the Relenza paper, which we didn't talk about, which looked at a couple of clinical studies in which they weren't controlled, but they're observational studies. And this is called, the paper was called Oseltamivir, the real world data, because that's like, in really sick people, does this help? And the bottom line is there's some evidence that it helps in really sick people, but again, the conclusions are that the trials aren't properly done. They're not extensive enough for us to really make a conclusion over whether this is good for helping really, really sick people. Now, one of the things that is quoted by Igor, he says it's meaningfully effective at preventing the spread of the infection. I don't know any data that addresses that. Maybe in cell culture, but I don't know any data that says it prevents spread of infection in people. Okay, so all I know is that there are some data about really sick people, but it's not terribly convincing. So, I don't know what the answer is. For healthy people, it doesn't seem to make much of a difference. The real question is, in a pandemic, when you're going to get serious illness, is it going to help? Maybe the only way to find out is in the next pandemic to use it, but you can't withhold it.
No.
It would have to be, as we discussed, an observational study. Okay, I don't know. I guess you have to. I don't know how you would do it. I don't know
you would get data on people who got it and who didn't for whatever reason, and
right.
And as they said in one of these blog posts, those kinds of studies are really influenced by bias. They can be really screwed up. So you have to be really careful. But that's the only way you can do it. So, I agree that there are some issues beyond what we talked about, and I appreciate. I learned that from reading these blog posts, and I think that's good. But I still don't think we have an answer about its usefulness in really sick people. With all due respect to Mark Crislip, his anecdotal feeling is not really useful.
Right. So it's possible that during a pandemic, when they start to use Tamiflu and they can't do it right away because they didn't know the disease was there until it was, and then they started to give it. Some countries might preempt the spread of that infection and give it as a preventative, and then you might find out whether it could prevent the spread of the disease or not.
That's what's going to happen.
That's what will happen, yeah.
Yeah, since countries have now stockpiled this stuff, assuming that they keep the stockpiles and they're okay. If there is, I mean, when's the next pandemic, right?
Right.
30 years?
Yeah, who knows?
Who knows?
I mean,
at any rate, there's likely to be, I can see where there's going to be observational studies of one sort or another following this up that will hopefully provide some insight into how these things work in really sick people.
I think also there are going to be better antivirals developed in the next 10 years, and we'll just forget about Tamiflu. But in the next 10 years, should we keep stockpiling it? That's the question.
How?
I don't know.
Well, the drug company certainly thinks you should.
Well, of course. Roche and GSK, is that the other company?
Yes. So there was discussion of data relevant to transmission in the Cochrane meta-analysis, right? So I recall the bottom line on that was that even though that was supposed to be part of some of the studies, that those studies were not good enough to give a conclusion. Is that right? Yeah,
that's why I said there's no evidence that it helps transmission from the meta-analysis, right? But I don't know why in that one blog post he said there's good evidence that it prevents transmission.
Right. That second blog post is by Scott Gavura, Gavura. Yeah.
So should we continue to stockpile Tamiflu? Does anyone have an opinion? I just don't know.
Sounds like we've already stockpiled a lot.
Yeah.
Is it enough? Have we stockpiled enough?
I don't know how many doses it is, I don't know. I just think it doesn't sound like a great antiviral. Of course, I'm not an ID physician, and I don't have trench experience in seeing people's lives saved by it. But who knows? Maybe they would have survived without it. I just don't know.
Right. Well, and what Mark Crislip kind of says is that, for the really sick patients in the hospital, this is when it's good. So have we stockpiled enough for that? I guess we don't know till we know how serious the next pandemic is and how many hospitalizations there are, but probably not everybody will be hospitalized with that level of illness.
Not a clear-cut answer, is it?
No.
Are there any? To any question.
Well, I think we should eradicate polio.
Right.
Yes.
Right.
Can we is another question.
That's right. Oh.
Dickson, let's do one more, and then we should move on.
Ricardo writes, "Hello, TWiV. Today, a newspaper politician opinion maker, which I usually don't like, due to differences in political spectrum, made a wonderful text about vaccination. Hopefully our country has a pretty good rate of vaccination, but as always, sometimes bad winds come to shore." As I said, it was a very well-written text, and he gives the reference to the text with the obvious
malinformed.
I'm sorry, malinformed.
It's one L.
That's right, malinformed comments from some readers, but the majority of them were with congratulations. In the middle of the text, it was a reference of a recent political measure made by Croatia about vaccination. And it's obviously they have a very high rate of vaccination, as Kathy tells us in her footnote. Maybe in the end, knowledge will prevail. That's a hope against hope. My best regards, Ricardo, who's an associate professor of microbiology, by the way, so he's a practicing microbe.
It's an old fan.
So I looked at this, and so it was in Portuguese, so I had to use Google Translator, which is not precise,
risky.
But I got the general idea, and the commentator is saying that the anti-vax thing is dangerous, okay, which makes Ricardo happy because he's ordinarily a fairly conservative commentator. I am getting the impression that anti-vax is slowly becoming, at least for the time being, socially unacceptable.
Correct. It's being marginalized.
Yes.
You're right.
Very interesting.
It's a Tea Party concept.
Even Jenny McCarthy recently is saying, I was never anti-vax.
Yeah, right. You want to get reelected?
Right. Really.
You want your pediatrician to stay with your kids?
And we've got, there was I don't know if we actually did, there was a Fox News thing that I saw on YouTube with a conservative Fox News commentator taking Jenny McCarthy to task on this.
Really?
And saying that being against vaccinations is crazy. I mean, what's happening here? This is nuts.
So, Kathy, you want to read your comment?
Well, just that this article that Rich talked about, translated from the Portuguese, refers to something in the Croatian Constitutional Court. And that article says that the court has upheld compulsory vaccination laws against 9 infectious diseases. The court statement has been translated as reading, "The child's right to health is more than the rights of parents to the (parentheses) wrong choice."
Bingo.
Yeah. And the Portuguese
crazy.
translated article cites that in Croatia, tuberculosis has decreased by 93%. Diphtheria, whooping cough, and measles have disappeared. 97% reduction in tetanus and 65% reduction in hepatitis B. There are things you don't want to lose. So, pointing out the efficacy of vaccines.
Yeah.
Well, we have a lot more email here, but I think we have to move on to picks. We'll have to do an all-email episode soon.
That's not a sexist remark, I hope.
How so?
No.
Email, M-A-L-E.
No, it's not at all.
Ha. I'm sorry, Alan isn't here right now, okay? So,
Dickson, do you have a pick?
I do. And I discovered it yesterday. And you can now download a live streaming broadcast from outer space by NASA from the International Space Agency, and you get their view out the window.
Cool.
It is amazing. You can sit there and watch the Earth rotate, or in this case, the spacecraft rotate. And you get the feeling that you're a part of the space station.
I see. You sent me the link here.
I did.
Live from space.
It's live from outer space. It's live. I mean, this is real.
So these guys can look at it.
Some guy wrote in a comment on this website that they wish that they could turn this into an automated website, a web saver, a screensaver. There's an ad that precedes it, of course. Sorry about that. You always have to put up with that stuff.
Yeah, it's like a half-minute ad.
I hate these things. I really
can't get through it.
I don't like it. But
let's see. There's a National Geographic link that might be faster. But.
Right. But it's amazing. I mean, if you ever wanted to go to outer space, here's your chance to know what it looks like from the window.
All right.
And a lot of us did that. So
thank you, Dickson.
You're welcome.
My wife is going to love this. She's
Good.
Yeah.
It's fun. I mean, it's a real-time thing, too. That's the part that makes it real, and not just like watching an aquarium that's been recorded and put up as a screensaver.
Kathy, what do you have for us?
I picked an interview that Tess Vigeland did, interviewing Alan Alda. And I can't remember if we've discussed the Flame Challenge before
on TWiV.
Yeah. So Alan Alda, a couple years ago, put together this Flame Challenge, describing what is a flame. And then he's established this institute named after him at Stony Brook. And he's continued to put out these challenges, and they get judged. So the current challenge is, what is color? It's a little bit too late to put in your entry for that for this year, but what I found in this interview was just some more entertaining discussion between Tess Vigeland and Alan Alda, and a link to a really cool Vimeo, the winning Vimeo of the Flame Challenge. And it really describes what's going on in a flame in a way that makes it understandable, and I need to watch it a couple more times to really cement it in my head, but I thought it was just outstanding.
Cool, yeah, it's great. So I was at a meeting not too long ago where the dean of the journalism school at Stony Brook, he was at this meeting, right? Sat next to him. So he's been involved with Alda at this center, right? And he told us what he does. It's really cool. He gets scientists to these training sessions where you use acting techniques to improve your communication, and I would love to go to one of these. I said, we do these podcasts. Why don't you tell Mr. Alda about them? And I sent him, he said, send me a link. That would be great if Alda said, hey, these TWiV podcasts are wonderful. And
Sure.
I mean, that's what he wants us to do, communicate science. So why doesn't he celebrate?
Why doesn't he come on our show?
Some of those.
Tell him to come on our show.
I don't know how you get in touch with Alan Alda.
You just put out a general bulletin.
I could ask this dean of the J School,
Of
but
course.
I never heard from him again.
Of course.
Kathy, I watched this video. I thought it was great, and one of the things I really liked was how they, for molecular models, they use Legos.
Yes.
I thought that was terrific.
Huh.
Yeah.
I asked my father this question when I was a young boy, probably 12 or so. I said, "What's fire?" And he said his answer was glowing carbon. And to some extent, according to this video, that was an accurate answer.
Wow.
Pretty good.
Rich, what do you have for us?
All right. Well, I was not with you guys last week because I was at Disney with my son and his family, including the 2 grandchildren. And in celebration of that, and because I think they really need a TWiV bump, my pick is Epcot.
Yeah.
Cool.
I love Epcot.
Yeah.
So
me too.
Epcot, this came to me in a flash. Epcot stands for the Experimental Prototype Community of Tomorrow. And we went around in the ride in Spaceship Earth.
Yes.
That's the history of communication.
That's great.
And when I came, I haven't been to Epcot for 20 years. And when I came out of that, I thought to myself, you know what? This is science outreach.
Yeah.
There are a whole bunch of people who ride these rides,
Of course.
who probably, there's a lot of information here that they probably don't have presented in a very digestible and enjoyable fashion, and to make them think about science and technology. And that's what Epcot is all about. We talk about outreach here all the time, and this is outreach in spades. So I just came away with all goosebumps about this, I just wanted to applaud that effort.
Richard, you know the advantage of being a grandfather, don't you? You get to relive your childhood.
I love this place. I love it. Actually, I was there, Rich, a couple of weeks ago when I was out for a week. I was actually there with my family, but I didn't tell anyone. And I love Epcot because there's lots of science outreach. In fact, Bill Nye is there sometimes.
Sure.
I said to my kids, I want to come here and set up something. Let them set me up, and I'll talk about infectious disease. And my kids are like, Dad, it's no good. You have to be more broad in science. They don't care about this. So that was really supportive.
The Epcot globe looks like a big virus. Tell them that.
Yeah, right.
Yeah, it does.
So I know a guy who worked in there. His name is Tim Blank, and I mentioned him on one of the other episodes as a pick of the week, Tower Gardens. He actually developed them as a result of working at Epcot.
Well, there's that exhibit called The Land. Where
That's the other one we went on.
Sure.
I thought of you the whole time.
Sure.
Yeah. In fact, Dickson, the last time I was there 2 years ago, I actually videotaped the whole land for you, but I never showed it to you. Have you ever seen it?
No, I have not.
It's very good.
You'd love it. You should go on this.
I'm sure. I will.
It's a history of agriculture, and then they take you through some experimental gardens, some hydroponic gardens.
Lovely. Yeah, no, that's right. So Tim Blank, that's where he got his big inspiration from.
My pick is a book that I recently looked at. Now, as chair of communications at ASM, I get books sent to me as they're published to check out. So this is cool. And this one I really enjoyed. It's called Medical Biotechnology. It's meant to be a textbook for, a course on that subject. It's rather long, but it's really got a nice summary of many areas. It's got some basic molecular biology. It's got some immunology. And then there's some recombinant DNA so you can understand biotechnology. And then they have chapters on gene therapy, producing therapeutic products in cells. It's a collection of things I have never found in one place.
Cool.
So for me, I'm developing a lecture on viral gene therapy, and this is a perfect resource for me. So I think if you're teaching a course on this, and there are plenty of courses on medical biotechnology, it would be a good resource for you. And it's not badly priced for a textbook. It's 700 pages, color, and it's only $120. It's not too bad.
Those of us who are going to ASM can check it out in person in the ASM book store.
Yes. Okay. They have a store there at the meeting, and you can go and thumb through it. I particularly like this.
Terrific.
So that's my pick of the week, and we have 2 picks from listeners. Nicola, greetings to the TWiV verse. I have a listener pick of the week. It's a microscope made from 50 cents worth of material, and links to an article about the microscope and its inventor, a Stanford bioengineer. Hope you can include this pick soon, as the group is running a contest to select 10,000 beta testers, and she gives a link to that. And I think this is pretty cool, so I wanted to give it a TWiV bump. Nicola in Connecticut, where it was a rainy spring day, 43F6C. P.S. As my name is feminine or masculine, depending on the country, I'd like to mention that I'm a gal. You know, I had a student named Nicola, and he was from Italy. So in Italy, it's a guy's name. Interesting.
Yep.
The other one is from John, who writes, Dear TWiV Superheroes. Here's a listener pick. Camtasia Studio is a computer program that will record your computer screen, microphone, webcam, etc. It lets you turn a talk or practice talk into a YouTube video with little effort. Going to a conference is fun, but having your efforts preparing a talk get stuck behind a paywall afterwards or lost completely seems completely senseless. Vince likes recording lectures. Maybe he can tell us what the optimal program is for such things, preferably one that doesn't have much of a learning curve. So actually, I have made a video on how I record my lectures. I don't use Camtasia because it doesn't give me the flexibility that I want. I use a program called Screencast. I use it to record all my videos. And sometimes when I go give a talk, I'll record my talk as well. And I have a link to the video, which I will put in the show notes. So thanks for that. This episode of TWiV will be at twiv.tv, also at iTunes. And if you like TWiV, go rate us on iTunes to keep us more visible. And send us your questions and comments to TWiV at twiv.tv. Dickson Despommier is at verticalfarm.com, trichinella.org, medicalecology.com, and urbanag.ws.
This is a test for Alzheimer's.
Thank you, Dickson.
Oh, it's a pleasure.
Aren't you going to pick your new podcast one of these days?
Perhaps.
You could be self-serving.
We'll get to that.
Yeah, we'll get to that. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun. I learned a lot.
Indeed.
Yeah, Dickson, you learned a lot.
Of more than they did.
And I'm going to see you, Kathy, in about a week.
Yep.
Cool.
Huh.
And you're going to meet Alan Dove, right?
Yep.
Nice.
In person. Yep.
But you've never met me.
No, but we'll do that too.
We're going to do it.
Do that in August.
We're going to do it in August.
If you show up, Dickson.
I'll be there.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Great time.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.